drugs, chloramphenicol, aspirin, etc., have got the hydroxyl or carboxylic acid group, and not only drugs but endogenous substances like bilirubin, steroidal hormones, and thyroxine utilize the glucuronic acid pathway for the excretion. See, one thing you have to remember that it may be an endogenous substance like acetylcholine. Endogenous means it is generated in the body. So endo means inside the body it is generated. Examples are hundreds of examples are we can available: acetylcholine, norepinephrine, many of the hormones, and bilirubin, steroidal hormones, and thyroxine, etc. They need to be excreted through the body, and they utilize the conjugation reaction to get out of the body. And drug glucuronides excreted in the bile can be hydrolyzed by bacteria in the gut. The liberated drug is reabsorbed and undergoes the same fate. So many times the lipophilic drugs, they get what you call as excreted through the bile. They undergo the enterohepatic circulation, and in the liver they are excreted in bile. They are excreted in bile, because then since they are still lipophilic in nature, they get reabsorbed into the circulation, and hence the half-life becomes long. What is half-life? Suppose a drug in the body is need to excreted throughout its what you call as the concentration becomes 0. Suppose 100 milligram is there, then it need to get excreted after 1 or 2 or 3 or 4 hours to the extent of 100%. So the elimination half-life is the one in which this 50% drug is excreted out of the body, or 50% is remaining in the body. So this enterohepatic cycling means absorption of the drug into the body, then it is going to the liver, then get excreted in the bile, once again getting reabsorbed into the circulation. So this example is phenolphthalein dye and oral contraceptives. Most of these undergo the enterohepatic cycling. So here the phase 2 reaction, drug should have, especially in the glucuronide conjugation, OH or CaOH group and glucuronic acid group. This should be present. That's the prerequisites to undergo the phase 2 conjugation reaction. The hydroxyl group, this is the hydroxyl group, or the acidic group, and the glucuronic acid, this is the glucuronic acid combination should be there. Here you can see some of the examples in which the metabolism of phenytoin by phase 1 cytochrome 450 enzyme, and the phase 2 uridine diphosphate glucuronyl transferase, UGT, it's also known as UGT. Here in many times what happens, if a drug is administered, then first it is going to undergo the what you call as the phase 1 reaction, maybe oxidation, reduction, and hydrolysis, you know very well. So the what you call as the product of that particular metabolism becomes substrate for phase 2 metabolism or synthetic reactions. So here you can see that phenytoin is one of the drug which will undergo both of these processes. So this phenytoin, or it is an anticonvulsant drug or anti-epileptic drug, it is highly lipophilic in nature. Why the most of the drugs like phenytoin etc. are lipophilic in nature is because they have to pass the blood-brain barrier. You know what is a blood-brain barrier? It's a thick capillary endothelium cell surrounded by the neuronal astrocytes and will not allow the drug to pass through the endothelial cells because they got the very tight junctions. So the hydrophilic molecules only can pass through the blood-brain barrier and the blood-placental barrier, etc. So phenytoin is the drug which is having the penetration capacity to the blood-brain barrier and going to act on the receptors in the central nervous system. So as it's very lipophilic in nature, the SIPE, so many varieties of SIPE already you have learned. the cytochrome P450 enzymes, it is converted to the 4-hydroxy-phenytoin, means in the fourth position here, 1, 2, 3, 4, the one hydroxyl molecule is added by the phase 1 reaction, and this becomes very slightly soluble in water. You got the point. In the phase 1 reaction, this is slightly soluble, is made as phenytoin. 4-hydroxyphenytoin is the one of the metabolite after phase 1 reaction, is slightly soluble in water. Whereas in presence of this UGT, which is also known as the uridine diphosphate glucuronyl transferate, uridine diphosphate glucuronyl transferate, or UGT. So this is a synthetic reaction now. It's a endogenous substances, and the UGT is combined with that. This is a UGT, and because of this thing, this 4-hydroxyphenytoin beta D glucuronide will happen. This one is very water soluble, and this metabolite is getting out of the body through urine. So usually many of the drugs also do, especially the lipophilic drugs undergo such type of the reaction. They will undergo both phase 1 and phase 2 reactions especially. So phenytoin in presence of CYP enzymes converted to hydroxyphenytoin, then in presence of UGT it's converted to 4-hydroxyphenytoin beta D-glucuronide, which is highly lipophilic in hydrophilic in nature and get excreted through kidney. And the second mechanism, apart from this glucuronic acid, the acetylation, means the compounds having amino or hydrazine residues are conjugated with the help of acetyl coenzyme A. Acetyl coenzyme A. So the groups, especially the drugs, if you are very expert in the chemistry, then if you see the structure of the drug, and you can easily say to which metabolite pathway it will undergo, whether it will undergo the phase 1 reaction or phase 2. So the amino or hydrazine residues containing drugs, they are conjugated with the help of the acetyl coenzyme A, that's known as the acetylation. The example of this category which undergo acetylation are the sulfonamides, isoniazid, then hydrolysed, etc. So these drugs will undergo the acetylation process, then get excreted through the body. And multiple genes control the acetyl transferase, and rate of acetylation shows genetic polymorphism, and slow and fast acetylators are there in the body. Some of the animals, especially the cats and dogs, the dogs cannot acetylate, or sometimes, so they are either fast or slow metabolites, and this depends upon the animals and its genetic property of the particular animal. And the third category is especially in the phase 2 metabolism or synthetic reaction. It is called as the methylation. The amines and phenols can be methylated, that means methionine and cysteine acting as methyl donors here, methyl. So example, adrenaline. So in presence of phenylethanolamine N-methyl transferase, so methylation occurs and is converted to the next metabolite. And histamines and nicotinic acid. This is the examples which undergo the methylation. So for each process, please remember 1 or 2 drugs which undergo this process. So whenever you get a what you call as the 10 marks or 20 marks questions, then you need to write all these things: histamine and nicotinic acid. Then sulfate conjugation is the fourth category in the phase 2 metabolism. The phenolic compounds and steroids are sulfated by sulfokinases. So sulfokinase is an enzyme, and the sulfur molecule is donated by endogenous mechanism, and sulfate conjugation occurs. The examples are the chloramphenicol, the adrenal and sex steroids, they undergo the sulfate conjugation. See, the thing chloramphenicol we have heard it several times, because the chloramphenicol, so it has to undergo several reactions to get excreted out of the body. That's why it will also undergo sulfate conjugation and nitro reduction. Ultimately, it gets excreted. So many reactions are available in excreting the drug from the body. So drug is a foreign substance to the body. Ultimately, it need to excreted out of the body. And here you can see that acetaminophen or paracetamol is having O-glucuronide conjugation. This is one of the reactions. See, a drug, sometimes if it is a lipophilic drug, it cannot be get excreted with simple steps. So it may undergo both what you call as the phase 2 reaction only. Once again, it may undergo glucuronide conjugation and sulfate conjugation. Then ultimately it is made as water soluble or hydrophilic in nature, and finally get excreted out of the body. So here you can see the example of acetaminophen. It is O-glucuronide conjugation will occur. This is the O-glucuronide conjugation. Then in this O moiety, once again sulfate conjugation will happen, and ultimately this get excreted out of the body. Then another fifth one is the glycine conjugation. So glycine is an endogenous substance. It will conjugate with that of some drugs which do have the salicylates, and other drugs having carboxylic acid group, COOH group, are conjugated with glycine. But this is not a major pathway of the metabolism. So glycine, this conjugation pathway is there, but when compared to the first reaction or synthetic glucuronic acid conjugation, this is not so important. And the sixth reaction is glutathione. Glucuronide conjugation and glutathione conjugation are different. So glutathione conjugation, then it is forming a mercapturate, is normally a minor pathway. So however, it serves as inactivate highly reactive quinones or epoxide intermediates formed during the metabolism of certain drugs, example paracetamol. So you may think that paracetamol is a simple drug, but even though it's a very simple, when so to get the paracetamol out of the body, lot of reactions are involved. So it also the reaction is also involved is the glutathione conjugation. So when large amount of such metabolites are formed in poisoning after enzyme induction, especially when high concentration of this paracetamol is given. This happens especially in case of cats and dogs. Many owners, what they do is they will go to the counter of the pharmacist and get the paracetamol tablet for the fever. Whenever the dog is not eating some food and other thing, they themselves will diagnose it as a fever and put the phenacetin or the paracetamol tablet to get the fever reduced. So this leads in toxicity. So then large amount of such intermediates are formed, like epoxides and quinones in poisoning after enzyme induction. Glutathione supply falls short. Toxic adducts are formed with the tissue constitution and they will damage the tissue. So when large amount of these intermediates like quinone intermediates and epoxides intermediates so are formed, and they cause toxicity when the enzyme cannot handle, means whenever there is a toxicity, such reaction glutathione conjugation occurs. Then apart from this one, ribonucleoside or ribonucleoside synthesis is also taking place, or the nucleotide synthesis or ribonucleoside or nucleotide synthesis. So this is one of the important for activation of many purines like the cyclic cyclic AMP, etc. and pyrimidine anti-metabolites used in cancer chemotherapy. So many drugs, especially purine and pyrimidine anti-metabolites, means anti-metabolites. It will stop the metabolism of the growing cells, especially some anti-cancerous drugs are there, and they undergo this type of the reaction.